This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 287, recorded on April 13th, 2023. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
What is it, like 90 degrees down there?
No, we have 76, 24 degrees Celsius, mostly cloudy, and it's moving into Chamber of Commerce weather. We're moving out of the winter, spring, into full-blown spring.
So we have a weird day today in New York. It's 30 degrees Celsius.
Ooh.
It's like in the mid to high 80s. It's unbelievable.
Yeah.
It's unusual. Also joining us from St. Louis, Missouri, Petra Levin.
It's good to be here. I think we also have mid to high 80s, which is kind of flipped that South Carolina is warmer.
Yeah.
Cooler.
Yeah.
Cooler.
Yeah. It's the
Weird
ocean.
weather.
It must be the ocean.
Must be the ocean.
Yeah, you're right on the water, right, Michael?
Yeah.
Okay, good for you. New York is on the water as well.
I've heard that.
Yeah.
I feel like you're on an island. Even.
Yeah, we're on an island surrounded by water. I have that accent, water. And then if you go all the way to the bottom, then you could see the Atlantic Ocean. But that's far from here. And where I am now, when I was at Columbia, I still am, but I don't have an office any longer. My office looked over the Hudson River. It was the most fabulous view ever. And I always said, how could I ever leave this? And you know what? It wasn't hard because I'm here at the incubator and I love it. So if you have something to go to, you can always leave. That's the key to life.
And Vincent is celebrating an anniversary of sorts this week. He is going to record TWiM 1000 on Saturday. So congratulations, Vincent, on reaching that milestone.
Thank you.
Yes, congratulations.
That's quite something, isn't it?
It is indeed.
Because even if you do 2 episodes a week, it's going to take you 10 years to hit 1,000.
Right.
And, well, we've been going more than 10 years, but the pandemic accelerated our productivity. So it's I want to do episode 2001, and I can call it 2001, A Virus Odyssey. Right?
That's what you're waiting for.
So I need to live for another 10 years. That's the bottom line. So I'm eating healthy. I'm exercising. I just can't sleep. So I don't know if that's going to make an impact, but
it can impact.
Well,
you need to sleep.
A neuroscientist told me it didn't matter too much, but what are you going to do? Maybe after 1,000 is passed, I'll sleep better. You know, that's
true.
Kind of that's the key.
At least I don't have to write grants anymore. All right. Today we have, for your pleasure on this nice weather, a snippet and a paper. And Michael, all about power, right?
It's all about power. And the title of today's paper is Investigating Variability in Microbial Fuel Cells. And it was published in Applied and Environmental Microbiology in ASM Journal in March of 2023. And it's by Daniel David Lester, Sam Settle, Claire McCann, Elizabeth Suden-Heydrich, and they're at the School of Engineering at Newcastle University at Newcastle upon Tyne in the United Kingdom. So in scientific studies, replicas should replicate, and identical conditions should produce very similar results, which enable parameters to be tested, hypotheses to be tested, and then, of course, the results verified. That's the thing I learned when I was introduced to science in middle school. The premise of today's paper is where the authors use real-world mixed inocula to generate a complex community that has a specific purpose or intent in mind, very similar to what we do when we all transit the birth canal to begin the colonization process that we refer to as the development of our microbiomes, or when our parents simply say, "Welcome to the world, this young one." And the diversity within these real world microbial systems like the human microbiome and like what we're going to be talking about today, microbial fuel cells, is often pretty great. And when a subsample of this diversity is harvested, taken, collected, or even placed into a reactor vessel or onto a surface to create a biofilm, stochastic processes are at work, or so we don't get a YouTube flag, E. coli happens. Who gets there first? What and you know what E. coli is short for. It's a four-letter word that begins with S and ends in T.
I prefer the one that begins with P and ends in P.
Yes, that's true too. But that also gets probably a YouTube flag. So who gets there first microbially? What nutrients are available? The compositions of that community, the players, and we've been doing lots of papers looking at census and communities, meaning there's baked in variability in all of this when communities are started. What we'll learn as we walk through the author's data is that we all should be aware when using mixed inocula in replica reactors under replica conditions, different communities may emerge that are all capable of different metabolic outcomes. And as Ilio would often say on this program, that is pretty much the story that we're going to discover here today on TWiM. But I think as the authors offer us for us to consider, and I'll quote them here, to date, there has been very little research focusing on this, meaning the variability, or even reporting it, with most studies using duplicate or triplicate reactors in which the phenomenon is not fully observed. Publishing data in which replicas do not replicate will be an important and brave first step in the research into understanding this fundamental microbial process. But if you think about it, we've seen this sort of thing when we have reviewed many of the microbiome studies that we've considered on TWiM, where we've witnessed the human microbiome being used in animal studies or in specific pathogen-free animals being used to investigate a phenomenon and then moving their stool into notobiotic or germ-free humanized mice. And I'm most especially reminded that it took the Parkinson's Society to reanalyze data from multiple clinical trials to collectively make sense out of the role that the microbiome may or may not play in the disease progression of Parkinson's. But I digress there. So let's get back to the paper to understand what's really going on. So what do microbial electrochemical technologies do? They abbreviate that MET. Well, they can be used for a wide variety of purposes, from simply waste degradation to really fancy organic chemical production. Within each piece of this tech is a biologic electrode, which consists of an electroactive biofilm on a conductive inorganic substance. If you will, something that's going to take the electrons. Now, this electroactive biofilm harbors electrogenic bacteria. That simply means they generate an electrical potential, or electrons, where they serve as a catalyst for the transfer of the electrons, and then the anode, this inorganic thing, transfers the electrodes to the cathode, which is a short way of thinking about we have a battery. So this is what microbial fuel cells are all about. It's really a microbial battery where the current or electrons are produced. The bacteria simply metabolize or eat reduced compounds, whether they be simple organic molecules such as acetate, and then the electrons, rather than going into the microbe's membrane to generate its energy pathway, they dump their waste electrons into the anode by any number of mechanisms spanning from nanowires that we've talked about on TWiM, electron shuttle mediators, and I'll bet you can all name an electron shuttle mediator. And I'll let you think for a second, and I'll bet you can all name one. And if you say you can't, sure you can. It's NADH. It's the simplest electron transfer device.
You didn't give us a chance.
I know.
All right.
I think of it as the donor.
Well, it's with the H. It's NADH.
Yeah.
It's got the electron. It's NAD. That's the oxidized part.
Well, here's another one. Fe3. Fe3 is another one, right?
So many of them. So many.
Yeah.
There are so many.
I think we should make a shout out for EEC, also to EET, this extracellular electron transfer, to Derek Lovely and Hema Reguera.
Absolutely.
Who really did beautiful.
They pioneered the field.
They pioneered it in Geobacter. Really beautiful.
Yeah, I remember reading that first science paper those many years ago.
Yeah, and Hema is wonderful, actually, if we want to have something else on. She's fantastic.
Yes. So the electrons then travel or flow, which the physicists and canoe paddlers referred to as current, from the anode to the cathode via an external pathway. And the hydrogen ions, with the bacterium just throws over the side out of the cell, are transferred in solution from the anode to the cathode compartment. And this then completes the circuit and enables the production of current. So summing up, microbial fuel cells rely on this bioanode, this cooperation of giving electrons to this inorganic thing, and then generate electrons simply from eating stuff, whether it be acetate or, in this case, sewage. So the process is pretty straightforward when uncomplicated substrates like acetate are being consumed. Then known cultures of electrochemically active microbes, that they abbreviate EAMs, can have and have been used to create inocula to create these bioanodes. The most known and extensively studied are, of course, the ones that came out of Lovely's lab and others, and that's Geobacter sulforeducens and Shionella ondensis. And Geobacter is an anaerobic bacterium naturally found in soils where it reduces iron 3 oxide, where Shionella is a facultative bacterium capable of anaerobically reducing metals, nitrates to nitrites, and chromates. Now, here's the rub, and what will serve as the basis of today's paper. When the digestion of complex organics is required, like sewage, as are many of the intended applications of microbial electrochemical technologies, a pure culture biofilm containing only electrogens often doesn't cut it. You need help. Hence the introduction of these mixed consortia of bacteria where they're needed to complete either the fermentative or the hydrolytic reactions. Now, the engineering community has learned that through bioaugmentation of anodes with an enriched electroactive consortium in microbial fuel cells prior to the introduction of the raw wastewater, they can significantly reduce startup times, which to them is measured in current flow. But to you and I that, use a spec 20 to measure our growth rate, that's, how long does lag take? And they measure it by something called coulombic efficiency, which compares reactors with and without pre-colonized anodes. So what's this notion called coulombic efficiency? And here to appreciate this, we all need to learn a bit about batteries. Since we're all soon going to be having an electric car in our garages, we should all learn something about batteries so that we might be more informed when we buy our first electric.
Is that right? Everyone's going to have electric? No more gas guzzlers? That's it?
If you live in California, it's coming sooner than most. I already have one in my garage.
Yeah, we have one too.
Yeah, so it's coming, Vincent.
I'd rather have no cars. How about that?
Oh, that would be the best, but some of us can't.
But you live in New York, home of subways.
Well, I don't live here yet, but I'd like to, and then I would get rid of cars, yes. And when I want to go somewhere, I'd just rent one, right?
Yeah. And, in simpler terms, and remember, my Maxwell equation training was about 46 years ago when I took the physics person of electricity and magnetism when I was an undergrad. what Coulombic efficiency is the ratio of discharge capacity after the full charge and the charging capacity of the same cycle. And it's usually a fraction of less than 1. Even in physics, nothing is absolute. And so why does this happen? Well, this gets to the gist of our paper. It's due to electrolyte decomposition, material aging, ambient temperature, and different discharge current rates, which you and I know as simply anaplerotic mechanisms. Somebody is stealing the electrons.
I don't know.
Yes, you do. You know that
Anaplerotic.
you have to make more bacteria. So how do you do that? You just don't, make a bucket of ATP and hold it like it's in a bank.
Anaplerosis
you steal those electrons.
Yeah, what is the
replenishing? Active replenishing.
The anaplerotic mechanism is synthesis.
Yeah. Replenishing TCA cycle intermediates that have been extracted for biosynthesis.
Yes.
Yeah, viruses do that as well. Viruses extract things for lipid synthesis.
They have anaplerotic
and then
mechanisms.
they put in other things, like glutamine.
Yeah. So we know this electrolyte decomposition as metabolic conversion efficiencies. And we learned that all in metabolism. And what these engineers want is the electrons that go to the cathode rather than making it into DNA, RNA, or proteins, or as we're going to find out in the next paper, lipids. Right, Petra? We're going to learn about lipids in the next.
No, we're going to talk about cell wall and then something that's called lipid too, but really is a cell wall precursor.
Oh, okay.
It's confusing.
So in the introduction of their paper, we have our first paradigm destroyer. Or simply, you would have assumed that a pure culture would consistently result in optimal performance when you feed a solution of pure acetate. However, pure cultures of electrochemically active microbes, while they've been successfully used with this microbial electrotech to generate current, it's now recognized that mixed cultures tend to produce higher current densities. And the beauty of studying, if you will, a microbiome of a microbial fuel cell is that you can measure things instantaneously with something as simple as a voltmeter. You can measure things in real time. And for us to do it in the microbiome, we either have to do 16S, we have to do RNA-seq, we have to do all of these other things. So these engineers really have it great because they can use a potentiostat and a voltmeter to figure out what's going on in their community. So the other thing we need to know before we get to the meat of the paper, within wastewater, electrogenic bacteria. So this is just garden variety sewage, everyone flushes. The electrogenic bacteria are likely to be present in very low numbers at approximately 17 electrogens per milliliter, or 17 part per million, if you will, if you're measuring the concentration of bacteria in millions, or 0.0017% of the population. However, when you subject their culture reactor vessel, and they give a nice illustration of this in figure 13 of their paper, you can see how simple this sucker is to design. They generate current within a number of days from this very low number of bacteria, and they just build up on the anode. They establish the biofilm, and their numbers increase. Now, first, the problem is wastewater is very different than pure acetate, and everyone flushes different stuff. So this will generate a fair bit of stochastism, which is involved in this inoculation. Not every bucket of sewage is going to be the same, and the wastewater is going to also contain microbes. Yet we know this technique works. But the amount of current that they're able to measure, expressed as both peak current as reflected by the maximum amount of substrate that's converted to current at any one time, and the total current produced from all the substrate present, is also affected by competing bacteria that do not possess these extracellular electron transfer capabilities. What's nice about microbial fuel cells, again, it's because you can study all of this in real time just by putting in electrodes and measuring the current differential. So we can simply use a potentiostat to measure the current output that the reactor is generating in real time. And here again, the authors offer in their methods how the calculations are done. And if you're a little rusty on your physics and electricity and magnetism like I was, this is a great section. And having had to sit on a student's dissertation committee that was working on this, I had to relearn much of my college physics to help him on his dissertation. To describe this in terms that we can appreciate and imagine in the presence of things like methanogens, we know that sewage makes methane, specifically hydrogen trophic and acetoclastic methanogens. They eat hydrogen and convert acetate to methane. So that's not good. That's not going to make current. So they can cannibalize or result in electron losses, which is a drop in voltage, with the conversion of hydrogen or acetate into methane. Similarly, the presence of sulfur-reducing bacteria in the sewage can result in sulfate being reduced to hydrogen sulfide, which if deposited on the anode, we know that hydrogen sulfide is often a poison to many microorganisms as it is us. And denitrifying bacteria are also in sewage, and they also can eat some of the electrons that are being produced in these reactors. Bottom line in all of this is an increase in current represents growth rates of electrogen. Therefore, as we monitor the average increase in amps measured per second, and this can be taken from each reactor for the period of time as a rise in current, and the rise in current is reported as milliamps per second. And when you look at their graphs, and lastly, the point at which the current started to increase above the threshold that they're able to measure, which is about 0.02 amps per meter squared of anode, this then is considered when the bacterium comes out of lag, and it's recorded in days. So what these authors did in their study is they investigated the variability in 28 identical microbial fuel cells reactors operated under identical conditions, with their goal of being to identify the scale and level of variability observed as measured by something they could do in real time. Peak current produced and the coulombs recovered from the same feedstock and inocula. Then they did a second set of experiments that to see if this variability persists as they would serially transfer in 3 consecutive feed cycles, either using just pure acetate or an acetate wastewater mix. And the reactors were inoculated with biofilms then from previous high-performing reactors to ascertain whether high-performing and more consistent biofilms could be created. And why does all of this matter? Well, if you think about it, if you're not working in microbial fuel cells, you're charged with making a probiotic or a prebiotic that will facilitate the transformation of your gut from a pre-metabolic syndrome patient to a normal metabolic phenotype, the holy grail of the microbiome community. And these guys are teaching us what we need to be doing in those sorts of experiments. And so much of what they're offering for our consideration in this paper really is quite important for the entire field studying microbiomes. And they learned that in their first experiment, the 28 replica reactors under identical conditions at the same time, using the same wastewater, provided them with a similar acetate substrate as feed. The results showed that there was a high level of variability in both peak and total current produced, and a moderate level of variability in the time that the community came out of lag at which the current levels increase. And you can see this on figure 1. It looks like a spaghetti plot. And you can see that the 28 reactors are all over the place. And this variability occurred during the first batch run with the inoculation and persisted in subsequent runs, regardless of whether those runs included freshwater inocular, pre-grown electrogenic inocular, or no inocular at all. And the interpretation of these data offer that despite the reactor configurations being identical and internal resistances between the electrode being very similar, they were consequential differences in biofilm formation within these reactors. And the establishment of the bacterial community in these microbial fuel cells can be simply understood in terms of classic bacterial growth under favorable conditions. You know, the 4 stages: lag, exponential, stationary, and death. And they have all of this in spades in their graphs. And they also offer us analysis of the succession of the microbial communities by measuring through analysis of 16S sequencing that shows us that even with 3 feeding cycles, the community in the reactor shifts significantly from the inoculum. And so this perturbation is a natural process that we really need to think about. And they attempt to control for this variability. In the 3rd round of their experimental reactors, they showed high performance reactors were used to inoculate further fresh sterile reactors with lower performing reactors as a control. And what they saw is, again, we had variabilities. Even the reactors given the good inoculum were observed to initiate current quicker and generally have higher peak currents. So again, why is there this variability? And the simple answer takes us back 134 years to 1889, where we have the great land rush of Oklahoma. And this is where we had 50,000 people lined up. In the case of their reactors, we had millions of bacteria lined up, some electrogenic, some not, even in the enriched electrogenic. And what they are arguing is that the mixed electrogenic fermentation is dependent upon the flock that you're with. You know, who's in your local community, and they can contribute to your electrogenic contribution. So it's really important to consider everyone there, because it's really about who gets to that clean anode first. It's really a time to the target, and that's dependent upon many stochastic variables that we can't control yet, or we don't understand how to control. So summing up, the authors present data that offer us that replication with microbial fuel cell experiments is very important. They showed that even with duplicate or even triplicate reactors, natural variability in performance could confound whatever parameter was being tested. The authors believe that their results highlight the need for more research into the causes of this variabilities and strategies to reduce it, including how to scale and how this may impact not only microbial fuel cells, but this other work that I've alluded to. As I read this paper, I was immediately thinking of the other stuff that we have reviewed on TWiM. So that's the snippet.
So, Michael, these are going to one day be used to make usable power. Is that the idea?
No, they're principally going to be used to get rid of.
Okay.
They're not going to be generating gigawatts. They're effectively going to be used to generate good stuff. They're going to be used to treat sewage. They're going to be used to make chirally active chemicals. Because remember, these communities are lean, mean machines, and we have to figure out what to get out of them. There are more efficient ways to generate our power than using microbes. And the downside is all of this makes CO2. And we want to capture CO2. So if we can use one of these reactors to fix carbon, that's so much the better. to tie everything up. Because this century is going to be about carbon sequestration.
Okay, thank you, Michael. Petra, what do you have for us?
So today I have a paper that's kind of in my own personal sweet spot of things I'm interested in. One is bacterial growth rate, and the other is bacterial cell wall, cell envelope synthesis. It's entitled Growth Rate is Modulated by Monitoring Cell Wall Precursors in Bacillus subtilis. It's by Yingji Sun, Sylvia Herleman, and Ethan Garner from Harvard University, and it was published in February in Nature Microbiology. And I like this paper also because it talks about growth rate, and bacterial growth rate is one of the things we're taught first, how to measure bacterial growth in culture using optical density. They actually look at single cell growth rate, but the same idea. And there's some classic work from what we call the Copenhagen School, some done by actually our own Elio Schechter and Ola Mola and Niels Kjeldgaard in 1958. Where they actually did very careful experiments showing that nutrients, and this is in Salmonella, but it's true in Bacillus and it's true in E. coli, if you take wild type cells and you culture them depending on the richness of the media, the carbon, how many precursors you supply, what's in there, they grow faster. So if you put them in minimal salts, like just the right salts they need, some phosphate, you give them a nitrogen source that's like ammonia or something not super easy to make into proteins, and you give them a carbon source they don't like, they grow quite slowly. But if you add a better carbon source and give them amino acids so they don't have to make them, and other things, maybe nucleoside bases, they grow much more quickly. And the idea has been forever, based on this beautiful work on which it's Schechter et al. 1958. I think it's a Journal of General Microbiology, classic paper, is that cells, bacteria optimize their growth rate for the media. They grow sometimes people would suggest even they grow as fast as they can given their conditions. That's not necessarily true, though, because, for example, you can have a fantastic carbon source, but you're growing on a terrible nitrogen source, right? Cells are not going to make carbon-intensive a bunch of lipids if they can't make proteins. So they're homeostatic control mechanisms that ensure that you basically go to the lowest common denominator. So if you have a poor nitrogen source but a great carbon source, you're still going to grow slowly because you still have to make the proteins that will make the lipids out of that carbon, for example. And there's beautiful literature on all this regulation. There's a little molecule that we think we've mentioned called PP-GPP, guanosine tetraphosphate, guanosine pentaphosphate. Those are the two kind of versions of it. And in the old school, they called it magic spot because it would appear when you starved cells for amino acids. You could see this nucleotide appear as a spot on a thin layer chromatography. And PP-GPP's role is really to balance the nutrients. But the other thing cells have to balance is actually production of their own precursors for all these different things they have to make. They have to make DNA, RNA, protein, cell wall, peptidic glycan, lipids, all these things they have to make from the precursors. And you don't want to make a lot of, for example, peptidic glycan without making membrane. So you need to balance that inside the cell, right? You don't want to build a house without a foundation, right? You want to make sure you have all the parts kind of made in order. And you want to make sure that your rate of making these doesn't exceed the rate at which you can make the precursors. So you have to coordinate assembly of all the different structures. And this paper is looking at how one bacteria, really Bacillus subtilis, so this gram-positive model system, coordinates the availability of the precursor for cell wall synthesis with assembly of the cell wall, right? You have this machinery to assemble the cell wall, but that machinery can't work unless you give it precursors. So you want to slow down the machinery if you don't give it precursors. And the precursors for the bacterial cell wall, so it's peptidoglycan. And the nice thing about that name is it has two sugars, N-acetylglucosamine and N-acetylmuramic acid, that are linked together, so a disaccharide. And then on that is a little stem of 5 little, like a peptide chain. It's not synthesized in the ribosome, but it's like basically 5 amino acid variants. So you have 2 sugars with a little peptide stem. And that is bound to a carrier because it's that little precursor is charged. That carrier is a lipid to get across from the inside of the cell where the precursor is made flipped outside. And the whole precursor is called lipid 2, really because of this carrier. Essentially, you need these precursors, and they have to be made in the cytoplasm and then flipped to the periplasm. And the cell has to sense in the periplasm whether it's getting those precursors. And the system for doing this, for making the cell wall, so there are two systems. There's one for division that we're not going to talk about. There's one for elongation. So Bacillus is a rod-shaped cell, and they grow lengthwise. And so they call it the rod system because mutants in the rod system result in round cells. So rod mutants are mutants that can't form round cells. And the rod complex has a couple different parts of it that are really important. So the first thing is there's a transglycosylase, rod A, which links together the disaccharide part of the precursor to form long chains of sugars or glycans. That's the transglycosylase. There's a transpeptidase that links together the peptides so that you've got peptide linkages and sugar links. The transpeptidases are actually the target of beta-lactam antibiotics, so ampicillin, penicillin. Those target and kill the transpeptidases. That's why they inhibit bacterial growth. We're not talking about those. I just thought that's something nice to know. So the rod complex is pretty big, and it has these transpeptidase, transglycosylase. It has on the inside of the cell a protein called MREB, which is an actin, sort of distant relative of actin. And it interacts to form these short polymers that can treadmill. And treadmilling is very interesting behavior. Again, not super important for this idea, but the little filaments of rod Z, one molecule would come off one end of it and be added to the other end of it. So you can imagine, I always think of Wallace and Gromit, the cartoons, if anyone knows those, where Gromit is trying to escape the evil penguin in the first one, and he's on a little train track, and he's putting down track as the train is moving. That kind of gives you a sense of treadmilling. So that's how I think of treadmilling. But these little MREB filaments, right, they are actually what's guiding the periplasm. So you've got on the inside of the cell MREB, and on the outside in the periplasm, or on the other side in Bacillus, which doesn't have a periplasm. So on one side of the plasma membrane, you have the MREB, and on the other side where the cell wall is built, you have these transpeptidases, transglycosylases. There's another protein called ROD-Z, and ROD-Z is actually kind of the connector. It talks to both parts, the cytoplasmic side and the extracellular side of this complex. So those are the things you need to know. Lipid 2, ROD-Z, the one that talks to the inside-outside, and then the synthetases, the glycosyl transferases, and the transpeptidases. Okay, so they're all linked together. The other thing that you need to know to understand this paper is a protein called MUR-AA, which actually is it is an enzyme that catalyzes the first committed step in making the lipid 2 precursor. If you have lots of MUR-AA, you make more precursor. If you have less, you make very little. And then there's one more player that we'll get to in this, but it's a serine threonine kinase. So it puts phosphates on proteins. In this case, it's going to put a phosphate on ROD-Z to regulate the activity of ROD-Z. And this kinase is called PRKC. It also has, PRKC has many things, but in this context, just ROD-Z. So it's a kinase that puts a phosphate on ROD-Z to regulate its activity. ROD-Z regulates the activity of the cell wall synthesis machinery for making the cells longer and rod-shaped. Okay, so this paper was interested in, first, how cells are able to grow more quickly. How do they make more cell wall in better media? And they knew from previous work that you have more rod complex activity, you have a higher density of these MREB filaments on the inside of the cell when cells are growing faster. When they're making more cell wall, you have more of these little filaments, which are holding on and guiding the cell wall synthesis machinery. So it makes perfect sense. So what they speculated was that there is some kind of sensor, some way for the cell to sense it's growing fast, or more directly, it doesn't sense growth rate, it senses the availability of its precursors, and it's somehow communicating that information to the rod machinery, right? the cell makes all this stuff in the cytoplasm. How does the rod synthesis, the longitudinal cell wall synthesis machinery, know when it has more or less precursors? So the first experiment, set of experiments, which is in figure 1, is they just kind of test to make sure that there is a higher density of these MREB. They actually use also another protein that's in that complex called MREC, higher density and richer media when cells are growing faster. And that is exactly what they see. It's a very nice set of experiments. If you grow cells in a rich media like LB, you see a lot higher density of these filaments. And they're using single molecule total internal reflection microscopy, which can actually look at single molecules on the surface of the cell. It's super nice. And they can see a higher density of these filaments in fast-growing cells. Okay, so that says that the cells that are growing faster do have more of this rod machinery, and they make more cell wall, which makes sense, because if you can't make cell wall, you're going to either get a big hole or you're just going to stop growing. So that makes perfect sense. Okay, the second thing they do, though, is they try to link this observation, which is very there's no experiment. It's really just seeing if faster growing cells have a higher density by actually changing the amount of lipid 2 precursor synthesis. And to do that, they use this enzyme I mentioned earlier, MURAA, which when you overexpress MURAA, you make more precursor, more lipid 2. And they find that if they increase the amount of lipid 2 by overexpressing MURAA, they get a higher density of the rod complex and cell wall synthesis is faster. What's amazing about this experiment, and I think really surprising, is when they make more lipid 2 precursor, they can get cells to grow faster, not in rich medium, but in minimal glycerol, which is kind of an intermediate condition for bacillus. Grows, I think it's like 50-minute mass doubling times in populations. It's not growing as fast as it can, but it's not growing as slow. Super slow. It's kind of in the middle. But when they increase, they can get 50, 60% faster growth rates just by increasing the amount of precursor synthesis, which is crazy because the whole idea is like they're optimizing their growth rate. Why aren't they growing fast in this media already, right? That was the whole basis for like our understanding of bacterial growth. And all you have to do is increase precursors and you grow faster. So that's kind of, I think, a super interesting observation. And this is all shown really beautifully in figure 2. And there's a really nice correlation between growth rate and lipid 2 and growth rate and density. Okay, so they want to know, though. Okay, so this was sort of their hypothesis that precursors were limiting. It's based on some previous work in E. coli and bacillus, but they want to actually understand the mechanism, right? We talk about mechanism all the time, and they want to understand the mechanism. So they only see this increase in growth rate in minimal media. They don't see it in the richer media conditions. So what they want to do is identify, they actually screen for drugs that inhibit growth rate in a media-specific manner. So they want to inhibit growth in minimal but not rich medium. The idea being that the sensor only functions, this mystic sensor they're looking for, only functions in nutrient-poor conditions because there's no difference when they overexpress the precursors in rich media. And they identify a compound called staurosporine, which is an inhibitor of serine-threonine kinases. So there's lots of different classes of kinases. This one is a serine-threonine kinase. That's where it puts the phosphate on the serine of client protein, the protein that gets the phosphate, it gets it on a serine. So there aren't that many serine-threonine kinases in bacteria. They're more common in eukaryotes, but there are 3 of them in bacillus. And they basically do genetics to identify one called PRKC, which has actually been characterized before. It's supposed to be involved in division, possibly DNA replication. It's been involved maybe in PPGBP regulation, lots of things. It's a membrane protein. It crosses, it has a cytoplasmic kinase domain, and it has 3 external penicillin binding protein domains. So it has domains that look actually like the transpeptidase, the part of the transpeptidase that I told you cross-links the little peptide stems to make peptidoglycan. It has domains that look like the active site of those proteins, which suggests that it might recognize precursor, which is like, I think, super exciting. And they find that previous work said that also implicates it in this potentially as the sensor is that PRKC activity increases in the presence of drugs that increase lipid 2 levels. So they have nice data that says it's PRKC. They then want to show it's PRKC. So they overexpress PRKC. Presumably, it's now hyperactivating its target, and it basically phenocopies. It looks like overexpressing MUR-A. It looks like when you make more lipid 2. So that works. If they overexpress it, they actually can increase growth rate up to 80% in minimal salts. Depends on the sugar. If they give maltose, which is not a great sugar, they get an 80% increase in growth rate. If they give it glucose, which is the best sugar, they don't get an increase in growth rate. So it really seems like it's tying it to carbon quality, which is really interesting, but kind of off another a tangent here. So I'm not going to deal with that. Okay, they want to know what PRKs sees. So also it increases MRV filament density. That makes sense. They grow faster, and you basically have more of these MREB filaments, which are required to guide the cell wall synthesis machinery. Okay, they want to know what its target is. The obvious target is actually ROD-Z. PRKC phosphorylates ROD-Z at serine 85. So ROD-Z, again, is this connector between the extracellular cell wall synthesis machinery and the cytoplasmic MREB. So it's been kind of implicated in that. And so really nicely, PRKC puts a phosphate on ROD-Z at serine 85. So they want to know, okay, is this needed? So they make 2 mutants. One is a phosphomimetic ROD-Z, S85E. So they put a glutamic acid there. And when you put a glutamic acid there, the things that it's called a phosphomimetic, it makes ROD-Z look like it's got that phosphate at serine 85. I love phosphomimetics. They also make a phosphodead. They take the serine 85, which would normally get the phosphate from PRKC, and they put an alanine there so it can't be phosphorylated. So they now have wild type ROD-Z. They have FOD-Z, the phosphomimetic, and the phosphodead. And the experiment that they do, which is in figure 4, is the phosphodead one that can't be phosphorylated, basically abolishes the faster growth that they see when they overexpress MUR-AA or PRKC. So that says you need to phosphorylate ROD-Z to sense the precursors. And the phosphomimetic S85E with the glutamic acid that looks like it's been phosphorylated, that one actually grows faster than wild type. So independent of PRKC and MUR-AA. It's just beautiful. It's like you can make the cells think that they have more precursor than they do by making the phosphomimetic, and you can make the cells blind to the extra precursor by making the phosphodead ROD-Z. What's also interesting, though, is the phosphomimetic, the sort of hyperactive one. That one, if you overexpress it too much, the cells lyse, because presumably they're trying to make cell wall and they're running out of precursors. And so their cell wall and their cell envelope integrity is lost. So why do this? They're not optimizing growth like we thought under all these conditions. They think maybe it's to ensure basically that pathways are balanced. So PRKC might tune the rate of lipid 2 production and PG synthesis across growth rates or nutrient imposed growth rates to ensure that you're not making cell wall at the expense of other pathways, that cell growth doesn't outpace lipid 2 production. Again, though, you can certainly grow faster, and the cells look pretty good when you overexpress MUR-A and PRKC. So, there's no reason they couldn't be making more precursor. So it's unclear exactly what's going on. I think one of the coolest other observations, or just so many kind of things to think about in this paper, is that when they increase growth rate by overexpressing PRKC or MUR-AA, the cells grow faster, and they do things that cells do when they grow faster. They make more ribosomes to make more protein. They make DNA faster. It looks like they initiate new rounds of replication more. They divide more. They basically have the impression, even though they're growing in a not great sugar like maltose or something, or glycerol, that they're growing in a better sugar. And so this idea, going back to this wonderful paper from Elio from 1958 and Olamola and Niels Kjellgaard was that the cells are really care about their nutrients. Here, it's like they care about their nutrients, but they're growing slower than the nutrient supply would suggest. So they haven't really found too much of a downside to essentially overproducing the precursors. They might seem to be maybe a little more sensitive to cell wall active antibiotics, but it seems like the cells are curtailing their growth, and it's, at least in the lab, it's unclear why that they might be doing that. So I thought that was just kind of super cool, and I liked that it connected us back to Elio. So
would you this expected to be in most gram positives, Petra?
PRKC is in the firmicutes, so staph has it. A lot of gram positives have it. Cool. RADZ is across the bacteria, but PRKC is really only a gram positive.
Okay. I think it's cool how you can take something from many years ago and keep learning about
pushing
it.
It's
just great.
I know it is. It's wonderful. I mean, this, it's fun to think about growth rate and what are the bacteria doing, and this idea that we think they're just optimizing their growth rate. I mean, clearly that's not what they're doing.
Yeah, right.
It's more subtle and interesting.
All right, that's TWiM 287. Show notes, microbe.tv/TWiM. Send your questions, comments to TWiM@microbe.tv. We have a few emails here. We'll do them next time. Petra has to leave now. And if you like our work, consider supporting us, microbe.tv/contribute. Michael Schmidt at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
And Petra Levin's at Washington University. Thanks, Petra.
Thank you so much.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.